I'm postdoc in Ryan Lister's lab here at UWA, and the whole lab is interested in based on epigenetics, and one of the important parts of that is DNA methylation. And DNA methylation is this is the presence of certain methyl groups in the 5' position on cytosines within the genomic DNA, and it is present in all eukaryotic organisms, and is essential for the structure of the genome, is transcriptional regulation. It is involved in imprinting, X chromosome inactivation, and for most tissues we have the methylation already just present in the CG denucleide context. So about 80% of CGs are methylated, for example, in the human genome, but in the brain, which is the organ I'm studying, we have actually another kind of non-CG methylation, CH methylation, where CH can be any other nucleotide, so A, T, or C, which actually is the dominant form of methylation in the brain. And we know from previous studies that this methylation in the brain is really important for the plasticity that is needed for learning and memory. There's also a huge overlap of key players in the methylation with the spectrum of autism disorders. And another prominent example for human disease is the mutation of the methylation reader MECP2, which results in Rett syndrome. It is also worth noting that the more complex the organism becomes, the more it makes use of DNA methylation as a regulatory mechanism. Now, this neuron-specific CH methylation is actually not present until birth. After eye opening in mammals, the CH methylation, which is shown here in blue, actually arises, and it takes until adulthood until it basically peaks and reaches this final peaking in its final form. And this is at the same time where also synaptogenesis is happening in the brain, where these neurons are forming connections to each other. And that's why it is thought that this DNA methylation in the non-CG context is actually really important for neural maturation. It was also suggested that it can be actually used as a marker for neural maturation. Now, to really be able to understand and study how this CH methylation is established and what the function is, we would need a model system, something that can easily be manipulated and observed. So ideally an in vitro system that allows us to investigate an in vitro system that shows CH methylation. And I'm using different human neural cell culture systems. I'm having, for example, classical 2D cell culture systems where cells grow where neurons growing in a monolayer starting from neural stem cells, but I'm also using cerebral organoids where they grow in a three-dimensional system. And knowing from human brain studies that, for example, Ryan during his postdoc in 2013 published, where he really did base resolution maps of the methlomes of brains in mice and men. We know that in adult human, this methylation can reach up to close to 10% in the CA context in this example. So I was well surprised and also disappointed by when I looked at these different cell culture systems that they nearly none of the CH methylation is present. And I looked at different cell lines. I looked at 2D and 3D. I enriched for neurons using either nuclear tags or sorting for nu n. I also looked at samples provided by other labs, and some of these samples have been cultivated for years. And although there is maybe a small increase happening over extended culture times, it is really very close to the detection limit of the methodology. So in human neurons in vitro, this does not to be, it seems that this CH methylation is not really turned on. So we turn to the other model system we have, which is using mouse cell culture system. And we initially wanted just to see if it is at all possible in vitro to get CH methylation by just prepping primary neurons from at E18 and cultivating them in vitro for about 2 weeks and checking in between if methylation shows up. We did this in 2 ways, 2 different ways. First, we used an antibody that was raised against the CA dinucleotide, methylated CA, and we actually can see that in neural precursors, we already see some of the CH methylation showing up, and later in new N-positive neurons, we see an increase of that. And then we also looked using a molecular method, that is whole genome bisulfite sequencing, where we made base resolution maps of this methylated positions in the genome. And here we see as well that within 2 weeks, so in a similar time frame that happens in vivo, if we take these previously these cultures, that these cells that initially have no CH methylation at birth, it can actually generate methylation in culture. So the next step was to look how does it look, if it is possible to get CH methylation, also when we use a system that is completely in vitro, starting with embryonic stem cells. So we used a standard protocol differentiating these embryonic stem cells using N2B27 media to make cortical neurons. And we again looked with antibody immunohistochemistry against methylated CA, where we can again see a signal, and also looking at different time points for methylation. And indeed, we see that around day 30 now of inducing the differentiation, we see this CH methylation starting to accumulate, and it's peaking at day 38 at a level very similar to what we see in adult mice in vivo. And this increase of CA around day 30, at the same time, we see an increase in the enzyme responsible for de novo methylation, which is DMT3A. So after confirming that these global levels are very similar between cell culture and at day 38 and in vivo neurons, we had a closer look how this methylation looks like in different contexts of the genome. So we looked at intergenic, but also genes and self, introns and exon at CGI islands around transcription start site. And we have on the left now CG methylation, and on the right we have the neuron-specific CH methylation. And this data is compared to the data that Ryan generated 2013 from in vivo data sets. And what we can actually see is that in cell culture, we actually have a hypermethylation. In our context, we have a little bit more of the methylation in both contexts than we actually find in vivo. But this methylation is not even, this surplus of methylation is not evenly distributed across the genome. We can see here, when we bin the genome into 100 kb buckets, we actually see that although few have actually lower methylation, the majority have higher methylation in CG. So, and this hypermethylation is actually so dominant that these two neuron populations, when you do hierarchical clustering, do not even cluster together anymore. For CH methylation, because fetal and glia nearly have none of such methylation, they do cluster together, but you can also see that there are massive differences in the distribution of these methylation markers. We also did calling DMR, so calculating which regions in the genome show differentially methylation. And this showed that while in intergenic regions we have actually less of such DMRs than you would expect by chance, we have an increase in methylation in both contexts in gene bodies, especially in exons. So the coding portions of the genome have the largest enrichment for such differentially methylated regions. We also compared it to developmental DMRs that Ryan generated back then. And this showed also an enrichment for fetal DMRs and a depletion for adult DMRs, meaning that most of the fetal DMRs that actually should become demethylated over maturation, they are still stay in the methylated stage, while most of the adult DMRs are actually not getting switched on. As we saw this enrichment, especially in the coding regions of the brain, we now had a closer look at the genes themselves and how the methylation in gene body looks like. So in this plot, we see, I plotted the methylation of all genes from the transcription start site to the transcription end site, including flanking regions, and plotting the methylation values, meaning here in red, more methylation, in blue, it's less methylation. And what you can actually see when you compare in vivo neurons to cell culture generated neurons, there is more red. So this is again the hypermethylation we see. And these genes have been sorted by the difference of methylation between these two neuronal populations, which can be seen here on the right. And you can see that about 90% of the genes show a higher methylation in vitro versus the 10% that show a lower methylation. We also did RNA-seq to compare it to expression of the relevant genes. Now, for CH methylation, it is actually a more 50-50 split. So about half the genes show a higher methylation in vitro and the other half show a lower methylation. And this, what you can see in this heat maps, especially this red, more darker red area is basically inverted in both neural populations, showing this split. And we again see that the highly methylated CH hypermethylated genes in cell culture are basically much less expressed than this is the case for in vivo neurons. And now when we're looking to get an idea about understanding which genes are mostly affected by the methylation differences, and by this hypermethylation, we perform gene set enrichment analysis to really focus on these parts. And what we see for CG methylation is that we actually see a lot of neuronal terms, so terms that we would expect to find in mature neurons, being like calcium signaling and receptor activation. We see cognition and learning, and a lot of channels showing up here. So these are terms essential for neuronal function, which seem to have the highest proportion of increase in methylation. For CH methylation, we actually don't see neuronal terms. We see more developmental head development, neural tube development. And if you compare them on a timeline of differentiation from the S cell to a mature neuron, these terms are coming up at CH methylation. They seem to be earlier during this differentiation, while the CG methylation terms are later along this methylation. So this kind of indicates that these two methylation programs are not kind of in sync, that we see differences in methylation earlier on in CH, and later on we see this difference in CG. What is also interesting to note is those genes that are lower methylated in vitro in the CH context. There's a lot of cell cycle genes being here, and it has been proposed that CH methylation is actually, when it is established depending on transcription. So genes that are initially highly expressed gain less CH methylation, and those that are lowly expressed gain more CH methylation. And this basically freezes this transcriptional state in a certain state for later on. So, and it seems, seeing that many cell cycle genes have a low methylation, it seems to indicate that when the CH methylation starts being established in cell culture, they are still in these, they still have these cell cycle genes active, which you would not expect to have in a post-mitotic neurons. So it seems that the CH methylation is actually established too early based on this data. And we also looked at similarity. So those genes that share actually a very similar configuration between in vivo and cell culture. And here again for CG, we actually see a lot of neuronal terms, while these are completely missing for CH. Again, indicating that CG methylation seems to be further down a maturation timeline than this is the case for CH methylation. So with all these differences, of course, you're wondering, is the CH methylation in these cell culture systems actually working properly? So, or is it a completely different state? So we compare the CH methylation in these cell culture neurons with ChIP-seq data from that were available from other studies in similar systems. So we confirmed, we compared the CH methylation and the CG methylation in our cell culture neurons to K27 trimethylation ChIP signal. This is a marker normally of heterochromatin that does not allow transcription, and also with K36 ME trimethylation, which correlates with transcription frequency. And this K36 trimethylation agrees with lower methylated parts, while the K27 trimethylation agrees with higher methylated parts. So this agrees with our CH methylation that we see. And based on our from our RNA-seq data, we also compared CH methylation along gene bodies with transcription, and it is known that both are anti-correlated in vivo. So higher methylation meaning lower transcription and vice versa. And the same is true for these cell culture generated neurons. Also, when we are going back to the staining of CA methylation in the nucleus, we observe that a lot of the signal of methylated CA is actually around the nuclear envelope. So this is perinuclear compartments in the chromatin that are also linked to a more repressive chromatin configuration. And we actually see the same pattern of staining when we look at cortex and hippocampus of mice that with the CA methylation that shows more of an envelope staining. So seeing that a lot of the expression seems to be in an immature state, we also looked at enhancer, and the consensus is that these enhancers, the CG methylation of enhancers in most cases will switch these off. So we were looking at a set of cortical enhancers from a paper by Shen et al. About more than 20,000 cortical enhancers that should be accessible in the cortex. And we see again in our cell culture neurons, these enhancers are indeed again hypermethylated. And then we look again at the genes that are linked to these hypermethylated enhancers. They are mostly enriched for really neural activity and synaptic terms and neurotransmitter release. We also did a motif enrichment of transcription factors that bind in these cortical enhancers, and this is a non-exhaustive list of those. So we're also looking what these transcription factors are, the expression and the methylation of those gene bodies, and we actually see it's a mixed bag. A whole bunch of transcription factors is lower expressed in cell culture, and as well having a higher CH methylation. And then we also have a whole group of transcription factors that's actually higher expressed in vitro and having a lower methylation. So overall, the whole state of the cell culture neurons that we can generate within time seem to be dysregulated. It does not represent an adult phenotype. So what does it tell us? In conclusion, we can say these cell culture neurons, and this is a problem we see with many cell culture neurons, and maybe maybe Gabriele today showed us a way out of this dilemma that most of the cell culture systems we have for human as well as for mouse show a fetal phenotype, and overall the neural configuration we have seen in this data set is also immature. And it seems based on low methylation status of cell cycle genes and the overall Go terms enriched comparing methylation patterns in the CH context, that CH methylation overall is established too early in these cell culture neurons, and that a lot of the hypermethylation we see might be actually based on that fetal DMRs are not being demethylated as they should, and also cortical enhancers not becoming demethylated, although the enzymes to do that are present. So overall, a lack of demethylation in many spots we looked at that might actually explain the hypermethylation we have seen. So can these cell culture systems still be used as a tool to investigate a methylome, especially the CH methylation? It is so far the only cell culture system I'm aware of that actually shows CH methylation, and a lot of the important hallmarks that we see from in vivo is present in the cell culture system. We have the anti-correlation with expression. We have the same nuclear localization by staining with antibodies against methylated CA, and we also see agreement with histone marks. So I would say the methylation itself can be used, and that the differences we actually see may be not related to the methylation itself. It is also not going back to our initial observation that in human neurons, we actually don't see any of the CH methylation at all. So there seems to be like a fundamental difference between human and mouse cell culture system And at the moment, we have no idea what this difference might be. If you are interested, we have a preprint available on bioRxiv. If you want to look up more details, you can also always select me an email if you have any questions. And as you can see from the author list, this is a collaboration. So I want to especially acknowledge Sally Martin and Ernst Wolfgang from the University of Queensland over East, with which we collaborated on this project, and also all the other authors and members of the team that were involved in this project. Thank you.
Thank you, Daniel.